Deep within the base of the brainstem lies the locus coeruleus. It is the brain's primary autonomous pacemaker, and from this tiny origin point, it extends a massive branching network of projections that reach nearly every part of the cortex. In the study of Alzheimer's disease, this structure presents a specific documented paradox. It is the very first region of the brain to accumulate toxic tau proteins. Yet by the end stages of the disease, it is also one of the most severely depopulated structures in the entire central nervous system. For decades, medical research linked those two facts together to form a single assumption: that the arrival of tau and the death of the cell represent one slow, continuous degenerative curve unfolding across a patient's lifespan. Look at this graph mapping disease progression. At break stage 0, a point in life when absolutely no tangles exist in the cerebral cortex, nearly 8% of locus coeruleus neurons already carry tau pathology. But as you can see, the cell counts remain perfectly flat and stable. The cells do not die. They survive in this state for decades. We have historically conflated a sick cell with a dying cell. The tau protein makes the locus coeruleus neuron sick, but for 25 years, it absolutely does not kill it. Unbiased stereology reveals another detail in this timeline. As the disease progresses through early stages, the overall volume of the locus coeruleus shrinks by 8.4% per Braak stage. But again, the physical number of cell bodies remains untouched through Braak stage 2. If the total volume is shrinking while the cell count stays exactly the same, the math dictates a specific physical reality. The neuron's massive, unmyelinated arbor is disappearing while its cell body remains entirely intact. If we expand this chart, we see that the locus coeruleus does not degenerate on a single timeline, but on 3 distinct clocks. First, a tau clock of sickness in the cell body. Second, an axon clock, where the projection networks collapse. And finally, a late-stage somatic clock, where the cell body actually dies. By focusing our imaging and our therapies exclusively on the first and third clocks, the initial tau and the final death, clinical medicine has entirely missed the middle clock, and that missing middle clock is where the actual therapeutic window lies. To understand how the arbor is lost, we have to look closely at what is happening inside the locus coeruleus cell body during break stage 0, when that early pretangle tau first appears. This tau does not sit inertly in the cytoplasm. It actively disassembles membrane-less organelles called nuclear speckles, which are critical command centers for the cell's genetic editing. When these speckles are broken apart, the cell suffers a severe depletion of essential scaffold proteins, specifically SRRM2 and pinin. Losing these proteins triggers a cascading failure in the neuron's RNA splicing machinery. The cell begins retaining introns, non-coding sequences of RNA that should be edited out. This transcription error occurs across hundreds of genes simultaneously. Clock 1 leaves us with a cell that is alive, countable, and still firing electrical signals, but completely stripped of its ability to reliably build complex isoform-specific proteins. The consequences of that internal failure do not show up in the cell body. They show up far away, out in the distant cortical target fields where the axon terminates, like the olfactory bulb. This split-screen chart illustrates data from an amyloid mouse model. On the left, we see a 33% loss of noradrenergic projecting fibers in the olfactory bulb by the time the mouse is 6 months old. But look at the control metric on the right. At 12 months of age, double the lifespan of the first measurement, these exact same mice show absolutely zero loss of locus coeruleus cell bodies. When we transition this chart to human tissue, the dissociation holds perfectly. Pronounced axonal fiber degeneration occurs at Braak stages I and II, hits a floor, and does not worsen as the disease progresses, all while the cell body counts remain flat and healthy above it. A full third of the brain's noradrenergic projection is physically erased from the cortex before a single parent cell body is lost. This raises the critical question: what is the second trigger? What converts a cell that has survived with tau for decades into one that rapidly loses its axon? The answer traces back to the splicing failure from clock 1. Locus coeruleus neurons pace themselves using a specific genetic break. The KCNIP4 gene encodes an A-type potassium channel break, which dictates exactly how fast the neuron is allowed to fire. Because of the RNA splicing failure, the neuron can no longer construct the specific K-CHIP4A isoform required to maintain that break. Without it, the pacemaker accelerates out of control, throwing the neuron into a state of hyperexcitability. The physical cost of this rapid, uncontrolled firing is a massive continuous influx of calcium into the neuron's thin, unmyelinated axon. This excessive calcium load forces a lipid called phosphatidylserine to physically flip from the inside of the axonal membrane to the outside. Once exposed on the outer leaflet, it acts as a highly visible "eat me" signal to the local immune system. Microglia, the brain's immune cells, read this flag using a bridging molecule and execute the instruction. They actively phagocytose or consume the living axon. The locus coeruleus axon does not passively wither away from starvation. It is actively eaten by the immune system because the neuron's own uncontrolled hyperactivity marks its projection as cellular debris. Moving to the absolute end of the arbor, we find the synapse is being dismantled by an entirely separate mechanism. A complement protein called C4D tags the synapse, instructing specific receptors to prune the connection away. A healthy neuron would normally sprout new connections to replace the loss, but in an Alzheimer's brain, amyloid elevates a molecule called effexin 5. This acts as a physical ratchet, permanently blocking the formation of new excitatory synapses. Once the connection is cut, it cannot be rebuilt. There is exactly one physiological state that halts this calcium-driven destruction. Is the architecture of human sleep. REM sleep is the only period in the 24-hour cycle where the locus coeruleus pacemaker shuts down completely to 0 Hz. This silence is the axon's only reprieve, the single interval where it stops generating the calcium flag that gets it eaten. Conversely, during NREM sleep, the locus coeruleus is required to fire in precise infraslow oscillations. These slow rhythms act as the mechanical pump driving glymphatic clearance, washing metabolic waste out of the brain. A hyperexcitable, diseased locus coeruleus cannot maintain this rhythm. It fragments sleep architecture entirely. The neuron loses its REM reprieve, guaranteeing the destruction of its axon, while simultaneously destroying the NREM pumping mechanism that clears toxins from the cortex. Removing the locus coeruleus axon initiates a systemic feedback loop that degrades the entire brain. Normally, the constant delivery of norepinephrine to the cortex acts as a standing inflammatory restraint. It keeps the brain's resident microglia calm and highly regulated. When clock 2 removes the axon at Braak stages 1 and 2, that noradrenergic restraint is withdrawn from the cortex decades early. Without that chemical suppression, the microglia become hyperactive and disinhibited. The diseased locus coeruleus acts as both the source of the fire and the operator of the sprinkler system. Its axonal retreat unleashes the exact immune cells that will go on to blindly destroy the brain's synaptic networks. Returning to our master timeline, we place clock 3. Cell body death finally drops at Braak stage 3. Notice this red marker, amnestic mild cognitive impairment, when a patient typically presents at a clinic, aligns directly with stages 3 and 4. Look at the massive horizontal gap before that clinical marker. That is a 25-year silent interval where the cells are sick but still alive. The interventions dictated by this specific biology are not aimed at saving cell bodies at the moment of diagnosis. The goal is deploying excitability brakes and protecting sleep architecture deep inside that 25-year window. By treating Alzheimer's disease as a single biological clock starting at diagnosis, we are trying to save an empty cell body, entirely missing the decades where we could have saved the projection that keeps the brain alive.